Hello students, welcome back. This is module 5.1.2, Origins Proto cells. In the last module we were discussing chemical evolution and the possibility of chemical self-replicating systems. And it is somewhat difficult to understand clearly how this would work, but just think of it in terms of those systems that did self-replicate, they would persist, and the ones that did not, they would not persist, and it's as simple as that. And our current topic is protocells, and protocells are the same things as protobionts, another name for them, and these are simple membrane-bound sacs that undergo some kind of chemical metabolism and are able to self-replicate. This is a hypothesized unit, and this is a unit that's thought to be transitional between chemical evolution and the first living cells. Now, we know that if we just take phospholipids and put them into water, that they will spontaneously form lipid bilayers. And it is believed that as part of the RNA world hypothesis, that RNA was the first information storing molecular system. Again, that's hypothesized. And we can ask ourselves, where might these first protocells have formed? And it turns out that there are many hypotheses. I'm going to give you two of them here. One is that it occurred, these could have formed on clay soils, maybe in tidal flats. Now, clay is soil of very small particles, and these particles are charged, and the charge of these particles could serve to stabilize molecules, such as amino acids and other things. And it is hypothesized a tidal flat because of the moistening and drying, and it would be at a particular spot in the tidal range so that it got just the right amount of moisture, and moisture and drying could lead to formation of amino acids and proteins. And that's one location. A second location that's hypothesized is down deep under the ocean and hydrothermal vents. Again, this is showing a black smoker here with sulfide chemicals coming out, sulfur chemicals coming out, I should say. And this is a close-up of some of the rocks, these little honeycomb rocks. They could serve as chambers that may have served as molds for the first cell membranes, and these little chambers could serve to isolate little self-replicating chemical systems, something along those lines. These are two of many hypotheses. We don't know where it occurred, but these are two possibilities. This is a model which shows the origin of the first cells. We start down here with number 1, and with the spontaneous formation of lipids, carbohydrates, amino acids, leading to proteins, nucleotides, as well, and this is under abiotic conditions. And Stanley Miller's experiment showed that this is possible. And by the way, I didn't mention at the time, but in addition to Stanley Miller, there's been a number of other researchers, and as our understanding of the chemical components of the early atmosphere have changed, as we've refined our understanding, so these different models have different experiments have used these different constituents. A second step is the formation of protein RNA systems, and independently, maybe we're having the formation of lipid spheres. And if we get that protein RNA system going, then there would be selection for a more stable molecule than RNA, and that would be perhaps the origin of DNA. And so we see DNA, RNA, leading to proteins. And then this could be a self-replicating system and enclosed in a selectively permeable protective lipid sphere. Well, this is pretty much a protocell, so the membrane protocell that has been hypothesized. And then, of course, it's a very small distance to go from there to living cells. Now, I know that many of these steps are somewhat improbable. Now, no one denies that, but the fact of the matter is they don't have to happen frequently. They had to happen only once over billions of years. And that is the materialistic explanation as to how life may have originated. And so this really is the best explanation that science has for how life originated. Remember, science can invoke a supernatural explanation and say, and then a miracle happens. But these steps are the possibility for how life materially formed. And once life was there, the earliest organisms were prokaryotes, or bacteria. And as we said at the beginning of the first module, fossils for sure 3.5 billion, maybe as old as 3.8 if we count those carbon specks. And the first bacteria were anaerobic. They didn't use oxygen. Later, prokaryotes became photosynthetic, and photosynthesis produced oxygen, changed the atmosphere, and this led to the development of aerobic respiration. Now, here's some photos. On the left, we see a fossil prokaryote, and on the right, we see a modern prokaryote. These are cyanobacteria capable of photosynthesis. And when they grow in little mats, and sediment can fall on top of the mat, and then the algae grows on top of the sediment, then more sediment, then more algae. So we get these alternating layers, and these form stromatolites. In this particular painting, these little things that look like cushions out here in the water, these are stromatolites. This is, again, showing some cyanobacteria. And this on the left is showing a eukaryotic form of algae from 2.1 billion years ago. Of course, you might be asking yourself, well, how did this 1972 penny, how was it present 2.1 billion years ago? But of course, this is a fossil. Ha ha ha. Okay, here are some stromatolites that are currently being formed. So this process is continuing. This is in Shark Bay in Australia, and this actually shows one of those little stone cushions sliced, and it actually shows the layers of, as I said, the alternating layers of algae and of stone or sediment that forms there. Anyway, this is going to complete this module as well as complete this first lecture on the origin of life. So thank you for your attention.